Okay, let's see if this works. Really quickly, I wanted to show you the lab that we did both on Monday and Tuesday. We did a stomach contents lab. And so part one is we decided to test the four major components of food. Those are simple sugars, starches, proteins, and fats. So what I wanted to do is show you kind of how we did this. So we started by running a positive and a negative control. In doing this, our negative control for each test is going to just be water. Water obviously isn't any of those four things. And so it's going to serve as sort of an example of what negative looks like. Now, for the positive tests, we've got kind of three reagents that we're going to use today. One is called Benedict's. Benedict's tests for simple sugars, meaning sugars that seem sweet. So we're going to use Benedict's to help us test for something we know has sugar in it, and then obviously with water, so we know what something that doesn't have sugar looks like. The second reagent we're going to use is called iodine. Iodine tests for the presence of starches. Starches are also sugars, but they're bigger, they're more complicated, and they're a good way to indicate whether a starch is present. So today I'm going to use something that I know has starch in it, and then obviously water is going to serve as my example of something that does not. The third thing we're going to test is protein. And so with protein, we use a substance called Biuret. Biuret, again, tests for the presence of a protein. So I've got something we're going to use that I know is a protein, and then, of course, water that does not. So for my sugar test, what we're going to do is we're going to test honey. So I made a honey solution up for the class, and I'm going to add it to one of my test tubes. The other one has just water in it. And to each one of these, I'm going to add Benedict's. So again, in the presence of a sugar, what we're going to do, or what we're going to see, is that Benedict's changes color. So I'm going to add one dropper full of the Benedict's to each one. And then for this reaction to take place, we're going to put them into some hot water. So I've got a hot water bath going right there. We'll come back and take a look at what the honey did versus the water in just a couple of minutes. Test number two is for the presence of starch. So in order to test for the presence of starch, I had to pick a food that I knew would test positive. So I chose some oatmeal. So I soaked some oatmeal in water and then kind of grabbed the water along with it. So I'm going to add one dropper full of my oatmeal mixture, maybe a little bit more here, along with my water. And in the presence of iodine, what we're going to see happen, we don't need to heat this one up. I'm going to add some iodine right here and right here. And what I'm hoping you should be able to see is that in the presence of iodine, anything that has starch in it, so that would be this one right here, turns kind of a dark black color. And in the absence of iodine, we have sort of just a gold color, which is the color that iodine is. So meaning there is no change in the color. Now the third test we're going to do is protein. So again, I've got something that is, we know, negative for protein, which is water. And to pull something that I know has protein in it, I'm going to use chicken broth. So I'm going to add just a little bit here to the bottom, hopefully kind of the same amount as I have water. Let me add a little bit of water here. So what we're going to do is we're going to add Biuret. Biuret tests for the presence of protein. So I'm going to add one dropper full to each tube. Again, I don't need to heat anything up when we do this. We simply just kind of look for a color change. So Biuret is a blue color. So what we see, let me just kind of swirl this up here for just a minute, is in the presence of protein. I'm going to add just a little bit more to make the colors show up a little bit better. Is a negative reaction for Biuret, meaning our water stayed kind of the same color. But the presence of protein, it turns kind of a lilac color. So I'll show you a picture of all of these at the end. So again, chicken broth tests positive for protein as I would expect it to, and water tested negative. Okay. All right, let's come back and just take a quick look at my Biuret, or my Benedicts, which is my sugar. Ooh, those are pretty hot. Let me pull these out of here with my cloth. You go ahead and turn my hot plate off as well. So what I was testing for was just simple sugars with that first test. That required a little bit of time in the hot plate. So what you can see here is that in the presence of a sugar, Benedicts will turn kind of a reddish orange color. Really what we look for is a color change. So it started off as blue, and I know that because water did not react. So it started off blue, and in the presence of some heat, and the Benedicts are honey, which is a very simple sweet sugar, turned orange, and my negative control, my water, turned, did not turn at all, so it stayed the same. So just to kind of show these to you one more time, let me kind of make sure this looks good. So kind of got a couple labels here for you to take a quick look. So what I have, just so I can show you kind of all of them side by side, my Benedicts, which tests for sugar, a positive reaction is an orangish red color, a negative is blue. So again, when a sugar is present, it will change to that red-orange color. When a starch is present, it will turn black in the presence of iodine, and it will remain kind of a goldish orange color in the absence of a starch. And with protein, we're going to get kind of a pink color when we have protein present, and it will remain blue, which again is the same color as the Biuret, in the absence of a protein.